Hello everyone, and welcome to this Labostix broadcast. This is a Labostix broadcast, part of the part of the broadcast edition, and this is a Labostix released a Labostix released part of the Labostix broadcast, and wherein this is a Labostix broadcast part of the Communicade, part of a Communicade. This is a chartered edition and an evidence-based perspective, and another Storm Labostix production. In this broadcast, there'll be an overview in around some works that been had been presented have been collaborative as collaborative some collaborative works between colleagues at an academic institution and wherein those works was also aligned to a particular a collaborative effort between a particular NHS a National Health Service trust in the United Kingdom. This works was this works was accepted for presentation and was presented in context of works aligned to within the remit of the UK Kidney Week 2022 and where this works where this collaborative works looked at a sort of a review in around pathology and analysis of approaches of easing kidney disease impact and looking at host pathogen host pathogen communication and biomedical visualization. This works was also collaborative in context in context of where there was a collaboration between the academic institution NHS Trust and aligned to the renal patient support group, the RPSG, as well. So here, as part of this Labostix broadcast, I thought I'd provide here. I thought it'd be a sort of opportunity, sort of, to share some of this works as I'm providing this sort of an overview. So to begin with some introduction. So since December 2019, the severe acute respiratory syndrome, the severe acute respiratory syndrome coronavirus 2 or SARS or CoV-2 had spread sort of worldwide, globally, causing COVID-19 to be problematic and implementing health risks across the globe. The coronaviruses at the same time generally can affect multiple organs in humans, for example, like the kidneys, increasing the mortality, especially in those who are immunosuppressed and or immunocompromised. Moreover, the lack of treatment forces the biomedical and pharmaceutical industries to seek approaches to offset healthcare crises. Where there is or has been a lack of effective treatments in around SARS or CoV or SARS and SARS or CoV-2, a more indirect approach should be considered to lower probable risk and onset of disease amongst immunocompromised and immunosuppressed individuals and patients. So the research surrounding COVID-19 has generally been progressive in context in around documented literature in around documentation and literature aligned to the public domain, and it is important to reduce all of the disease mortality at epidemiological and population level. However, the understanding of structure and pathology of coronaviruses, especially surrounding host-pathogen and communication and visualization, requires insight. So in around this works a little bit some bases in around sort of the biomedical classification in around as part of this intro. So the coronaviruses are perhaps more a recent discovery than of a more of a recent discovery in some regard, but at the same time, coronaviruses are those are viruses are viruses that are linked to adenoviruses that fall under the same category, and where retrospectively there has been more taxonomy investigated. The first coronavirus is strain B814 was discovered originally in around 1965 by scientist Tyrell Bino, and where biomedical specimens were collected from the respiratory tract of a patient with a common cold. Subsequently, samples were then linked directly to human volunteers to develop disease and evidencing the presence of infectious agents. Thereafter, works by Hamer and Procknow were then able to grow isolated virus strains, including B814 and 229E, which improved sort of the early biomedical visualization in context of extraordinary viruses. So in around sort of the aims and objectives of this collaborative works, the works aim to investigate causes and potential mechanism during severe acute respiratory syndrome or SARS and SARS and infection and CoV-2, the sort of renal interplay or interaction, and the effects of acute kidney injury or AKI. This works was also collaborative in the sense that a full a sort of a chapter contribution towards a particular book publication had been put forward as well, and so this works provides a review of the pathology and analysis of approaches to easing kidney disease impact by exploring the host pathogen communication and providing a biomedical visualization perspective, and highlighting the principles really of angiotensin converting enzyme 2 or ACE2 in context of immunology and infection to also understand the impact of kidney disease in around this. So on from this, a little bit around sort of the healthcare, sort of the complexities in context of healthcare. Um, so based on taxonomy and pathological features, a team of eight virologists decided to classify them classify classify them as coronaviruses due to the characteristic appearance. And ongoing research and microscopy advances surrounding coronaviruses provided greater knowledge of viral structure and function to discover therapeutic possibilities around unpredictable outbreaks. Advanced microscopy and visualization have been important throughout practices and updates surrounding the pandemic. Regarding the pharmaceutics and in context of angiotensin, in context and around angiotensin converting enzyme inhibitor 2 or ACE2, it has there has there's literature to inform there is a diverse interplay as the SARS-CoV-2 impounds respiratory health can impound respiratory health, and the implications prompt symptoms that have been highlighted in literature relating to COVID-19, the COVID-19 pandemic, and how related to the COVID-19 pandemic, and have become more obvious in following the SARS outbreak the SARS outbreak from the Wuhan province in China from 2003. So in following this as well, an analysis of coronaviruses in correlation with renal health, and sort of the novel therapeutic strategies relating to preventing symptomologies in around this will be outlined, and where the next sort of basis in around this works will specifically be in around the sort of biomedical visualization. So in context of the renal and COVID-19 correlation, it had been highlighted that the first pandemic, or the first pandemic caused by coronavirus SARS-CoV occurred in between 2002 and 2003 in Asia, where around 8,000 around 8,900 8,000, just over 8,000 patients, were evaluated for a positive for the virus, and as a result, 774 deaths had been documented. Despite common acute respiratory failure caused by diffused alveolar damage, multi-organ failure was noticeable, wherein there were cases of severe, for example, symptomologies aligned to diarrhea, hepatic failure, and acute kidney injury, or AKI. An investigator from Hong Kong, after evaluation, highlighted that 536 SARS 536 SARS of cases in around 6.7% of patients developed specifically acute kidney injury, or AKI. Moreover, the mortality amongst this group was 91.7%, which is significantly which was and is higher, had been higher than patients without any symptomologies of AKI, which was 8.8%. So the middle, in light to this as well, the Middle East respiratory coronavirus, or Middle East respiratory coronavirus, which has been documented as MERS-CoV, induced two epidemics, one in 2012 surrounding countries in the Middle East, and one in 2015 in South Korea. The MERS-CoV implicated the lives of 2,562, resulting in 881 deaths. To determine possible nephrological complications associated with MERS-CoV, investigators from South Korea examined 30 patients by monitoring vital signs, bacterial culture of blood, of bodily fluids, virus polymerase chain reactions, and within a testing group, proteinuria occurred in around 40% of cases. Hematuria occurred in just over 63% of cases, and acute AKI, or acute kidney injury, developed in around 26, just under 27%, 26.7% of patients, and mostly in the elderly population. And around this as well, and sort of specifically the biomedical visualization and analyses of COVID-19. The coronavirus is divided into four genera. There's so number one, alpha coronaviruses, H-CoV-229E, and beta coronaviruses, where there are variations of beta coronaviruses, where between alpha coronaviruses and beta coronaviruses, there then is then a differentiation where there's a differentiation between alpha coronaviruses and beta coronaviruses, which originate from bats, and gamma coronaviruses and delta coronaviruses can be transmitted from types of birds and swine, and swine gene pools. A SARS-CoV-2 virion contains an unsegmented single-stranded positive sense RNA genome of around 30 kilobases, which encodes in crucial viral protein used to create progeny viruses, and the four essential structural proteins called membrane, envelope, and nucleocapsid, and spike. As far as airborne viruses are concerned, SARS-CoV-2 is transmitted between humans due to entering the viral particles to the upper respiratory tract. The viral entry occurs by binding of the spike glycoproteins, which are homotrimers present on the surface of the coronaviruses and where the spike proteins are cleaved by host proteases into subunits. S1 subunit for receptor binding, and S2 subunit for membrane transfusion. Microscopically, the infected cells are forced to translate proteins required for generating the virus's progeny. In the original works, in the original works around this, there is a schematic which goes through the taxonomy and an overview of coronaviruses. Uh, 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 Again in the original collaborative works. Uh, in around laboratory practices, and where this where this is in around the remit of organ crosstalk and biomedical communication. So most informative methodology to analysis disruption caused by SARS-CoV-2 is SARS-CoV-2 is biopsy of renal tissue or kidney tissues. With the usage of light and electronic microscopy, samples overgo structural examination to identify injury within tubule, epithelial, and glomerular cells. Linked to this is polycytopathy, which in effect is a glomerular disorder and is mostly observed within COVID-19 patients, possibly caused by cytokine-mediated polycyte injury. Examinations also provide data of development of acute tubular injury, or ATI, as a predominant major effect of SARS-CoV-2 infection. Results of predisposed patients includes sort of evidence in around the crescentic transformation of long-standing pre-existing class 2 lupus nephritis, and development of acute T-cell mediated rejection in patients with transplanted antibodies, indicating possible injury caused by induced acute rejection, in context of of injury, where glomerular nephritis is concerned. Microscopy evaluation has demonstrated absence of virions with kidney cells in all patients. Immunohistochemical is selectively immunohistochemical is selective is selectively utilized identifying antigens in tissues and cells of sections by exploiting principal antibodies binding specific to antigens in biological tissues, which have shown a viral genome in kidney cells. For SARS-CoV-2, the stain for S2 subunit of its spike protein and nucleocapsids protein are performed with the usage of appropriate monoclonal antibodies. For all investigations, the results demonstrated absence of virions within kidney cells. Microscopy evaluation of kidneys in patients with COVID-19 provide a range of highlight a sort of a range of renal complications. This is a requirement for further. There is a requirement for further investigation to understand the data which could potentially provide clearer understanding of renal microscopy affected through COVID-19, or where COVID-19 is documented. Understanding now in around this and understanding sort of renin-angiotensin-aldosterone system pathway and the role of ACE2 during COVID-19. So the renin-angiotensin-aldosterone system is a multi-hormonal regulator of blood volume and systemic vascular resistance. Angiotensin is cleaved by renin forming inactive decapeptide Ang1, which is next converted by ACE into active octapeptide Ang2. The binding of Ang2 with adequate angiotensin receptor AT1 or AT2 leads to hypertension, inflammation, and multi-organ failure. Works by teams in around 2020 had highlighted common and potent antiviral drugs. However, before these, ACE2 inhibitors had been available to prompt blood flow between renal, afferent, and efferent arterioles, which subsequently action oxygenation of cells throughout general physiology. ACE2 performs more proactive, a more protective role by degrading Ang2 to active peptide Ang1 to 7 through cleavage of the carboxy terminal phenylalanine in Ang2 has been evident as part of the mechanism as well. Products products of ACE2 action binds with mitochondrial assembly protein 1 or MAS and receptor, leading to vasodilation, vasoprotection, and cardioprotection, showing regulatory roles of both receptors. However, ACE2 receptors are inhibited during SARS-CoV-2, which downregulates protective action and leads to severe development of COVID-19 by hypercoagulation and microangiopathy. The infected cells are forced to translate proteins required for generating the virus progeny, which also provokes the immune action. So in conclusion around this works, there have been a surge in literature surrounding mechanisms attributing to SARS-CoV and SARS-CoV-2 action on immune response to pathogen. SARS-CoV-2 patients present increased level of blood coagulation factors and blood clotting complexities, and the main mediators include thrombin, tissue factor, and fibrinogen. SARS-CoV-2 infection impairs the function of vascular endothelial function, resulting in increased production of thrombin and reduced fibrinolysin, which impacts clotting ability. Increased fibrinogen expression helps in platelet aggregation, which promotes clotting formation in blood vessels, resulting in dysregulation of cardiovascular impediment. Patients can develop myocarditis, arrhythmias, heart failure, acute coronary dysfunction, and or sudden death. There is an indirect advantage. There is an indirect advantage in implementing angiotensin converting enzyme 2 or ACE2 treatment to improve the immune response against infection. ACE2 may also offer appropriate strategies for the management of symptoms that relate SARS-CoV and SARS-CoV-2 in most immunocompromised or immunosuppressed patients. However, what is still not known is whether ACE2 should be ACE2 should be assigned at population level to totally prevent COVID-19 symptoms. That notwithstanding, advances in laboratory practice allow implementation of pathology assays and techniques and investigations, including microscopy advances for visualization and analyses to understand host pathogen communication to preclude kidney disease impact. That draws this Labostix broadcast to a close. There will be resources linked with this sort of overview in the descriptives in social media, so listeners out there can identify further information through referring to the resources through sort of approaching the resources this again, this works had been this collaborative works had been presented at the UK Kidney Week in 2022, as sort of aforementioned in the beginning parts of this Labostix broadcast, this work had contributed as a chapter towards as part of a chapter in as part of a book publication according to pathology as well. So thank you for tuning in, and there will be another Labostix broadcast in following. This has been a Labostix broadcast, part of a broadcast edition, a Labostix release as part of as well, and a broadcast edition aligned to the Communicade, a chartered edition, an evidence-based perspective, and another and this has been another Storm Labostix production. Thank you.